Hepatitis D virus, or HDV, is a strange character in the viral world. Unlike most viruses, it can't survive on its own. It needs help, a partner in crime. That partner is the Hepatitis B virus, or HBV. Without HBV, HDV is powerless, but together they can cause serious liver damage. HDV is found worldwide, but it's more common in certain regions like the Mediterranean, Middle East, and parts of Asia and South America. Why it clusters in these areas remains a mystery, a puzzle for viral detectives to solve. What makes HDV so interesting, so unique? It's a hybrid, a blend of different viral strategies. It acts like a satellite virus hitching a ride on HBV, yet it also has its own tricks, its own ways of wreaking havoc in the liver. Understanding HDV is crucial, not just for scientists, but for all of us. It's about protecting ourselves and future generations from this tiny but potent threat. To understand how HDV works, we need to zoom in, to peer into the microscopic world of viruses. Imagine a tiny, intricate machine, each part designed for a specific purpose.
At the heart of HDV lies its genome, its genetic blueprint. Unlike our DNA, which is a double helix, HDV uses RNA, a single strand of genetic code. This RNA strand is circular, forming a closed loop. It carries the instructions for building new viruses for replicating and spreading. Surrounding the RNA is a protective shell called the capsid. Think of it as a viral suit of armor. This capsid is made up of proteins, the building blocks of life. But these proteins aren't made by HDV itself. They're made by the liver cells that HDV infects, hijacked to do the virus's bidding.
The viral key. Embedded in the capsid is a special protein called the hepatitis D antigen, or HDAG. This protein is the key, allowing HDV to enter liver cells. There are two forms of HDAG small, or SHDAG, and large, or LHDAG. They work together, a tag team in the viral invasion. Borrowing a coat. HDV is a master of disguise. HDV doesn't have its own outer coat or envelope. Instead, it steals one from HBV. This stolen envelope is crucial. It allows HDV to attach to and enter liver cells, camouflaged as its partner in crime.
Section 3. How HDV spreads. HDV is a dependent virus, unable to survive without HBV. It's like a parasite, relying on its host for survival. This dependence makes HDV unique and limits its ability to spread. A dependent virus. HDV can only infect people who are already infected with HBV. It's like a two-step process. First, HBV infects the liver, then HDV can piggyback on HBV using its envelope to enter liver cells.
Pathways of infection. HDV spreads through blood and other bodily fluids. The most common routes of transmission are parenteral, which involves sharing needles or syringes contaminated with infected blood. Sexual, through unprotected sex with an infected person. Vertical, from an infected mother to her baby during childbirth. And finally, nosocomial, which is exposure to infected blood or needles in healthcare settings. Double trouble coinfection and superinfection. There are two ways HDV infection can occur. Coinfection, where a person gets infected with HBV and HDV at the same time. And superinfection, where a person who already has chronic HBV infection gets infected with HDV. Superinfection is usually more severe than co-infection, leading to more serious liver damage.
Section 4. HDV's attack on the liver. Once inside the liver, HDV launches a multi-pronged attack, damaging liver cells and disrupting normal function.
The body fights back. Our immune system tries to fight off HDV, but the virus is cunning. It can evade the immune response, hiding within liver cells. The immune system, in its attempt to clear the virus, can also damage healthy liver cells, adding to the problem. A two-pronged assault. HDV damages the liver in two main ways. First, there's immune-mediated liver injury. The immune system recognizes HDV-infected cells and attacks them. This immune response involves specialized cells like cytotoxic T cells and signaling molecules called cytokines. Second, there's the direct viral cytopathic effect. HDV can directly damage liver cells by interfering with their normal functions. This direct attack disrupts the liver's ability to filter toxins, produce proteins, and store energy.
The antigen's role. EITOLOGY, the viral key, plays a role in liver damage. It can trigger inflammation and cell death, contributing to the progression of liver disease.
Long-term consequences. Chronic HDV infection can have serious long-term consequences. Fibrosis. Scar tissue builds up in the liver, impairing its function. Cirrhosis. The liver becomes severely scarred and shrunken, leading to liver failure. Hepatocellular carcinoma, or HCC. The risk of developing liver cancer increases significantly with chronic HDV infection.
Section 5. Prevention is key. HDV is a serious threat, but it's a preventable one. The key to prevention is vaccination against HBV. The HBV vaccine is safe and effective. It protects against both HBV and HDV infection. When someone is vaccinated against HBV, they are also indirectly protected from HDV. Other preventive measures include avoiding contact with infected blood and bodily fluids, practicing safe sex, avoiding sharing needles, and getting tested for HBV and HDV if you are at risk. By understanding HDV, its structure, etiology, and pathology, we can make informed decisions to protect ourselves and others. Vaccination, along with other preventive measures, can help us win the fight against this unique and potentially devastating virus.